Well, hello everyone. I want to welcome you to today's seminars from coming to you from Yale and Harvard. It's kind of really a community feeling here today because Frank Slack is the founder of actually the Yale RNA Center. And I have the pleasure of being the director of the center. I'm the third director. And I really appreciate Frank's insight and energy that he put into establishing our center. It's really a wonderful community. And I'm so excited to introduce Siggy Nachtergaele today, who's one of our really energetic and insightful junior scientists at the center. She's an assistant professor in MCDB, and she's taken so many roles at the center. Most recently, she's been leading our postdoc program that we have at the RNA Center together with Junji Guo. And she really has the ability to impart wisdom and encouragement and a sense of optimism to our postdocs. So I want to say a few words about Siggy, of course. She did her PhD at Stanford University, working on development in Rajat Rohatgi's lab. I had to practice saying his name a few times. Sorry if I butchered that. And then after graduating in 2014, she went on to Chuan He's lab, where she was working for the first time on RNA, I think, and RNA modifications, a number of different ones. Studying her CV was a pleasure. I got an education in her background, which is remarkable. And I just want to make a couple of observations. I think Siggy's a fantastic scholar. She's written a beautiful annual reviews of genetics in 2018, a beautiful review that really brought my attention to her name initially on RNA modifications. But she's also an achiever on the one hand, obviously works really hard and is very insightful about direct scientific directions. She published two very impactful papers in Chuan He's lab in 2016 and 18. And these articles really gave us an idea of where RNA modifications were going and the types of activities they had in biology. And so this is really part of the wave of excitement that has come over us all. And the other amazing thing about her CV is how clear it is that she's a team player. She has many publications from both labs where she was middle author on a variety of topics that really seemed like she was helping others because they weren't exactly directly in her exact project. And so this actually matches the impressions that I have of Siggy, and I'm so grateful to have an involved junior faculty member in our community. So with I just did want to mention she's gotten a few acknowledgements of her excellence during her assistant professorship here at Yale. In 2021, for example, she got the Sontag Foundation Distinguished Scientist Award, and as well as two others. I think that these recognition opportunities do signal the things that we have to look forward to coming from Siggy's lab. So without further ado, I'll stop talking and allow her to tell you her wonderful results.
All right, thank you, Karla. So hello, everybody. I'm gonna, let's share, there we go. Move some things around here. So first of all, thank you so much, Karla, for that very kind introduction. I'm now gonna proceed to try to live up to that introduction, and we'll see how that goes. But I'm really excited to have this opportunity to share some of the work we've been doing in the lab recently. This is ongoing work that we are writing up at the moment, and we are really excited about it. So I'm really thrilled to share it with you. So the work that I'm going to be telling you about today has to do with how chemical modifications on mRNAs regulate stability of these RNAs in drug-resistant glioblastoma. And so I will just start by saying that, of course, RNA, the molecule we all love, I probably don't have to convince this audience that it is much more than just the intermediate molecule in the central dogma, and in fact has a lot of really interesting regulatory functions in and of itself in cells. And what our lab really focuses on is sort of how these RNAs are regulated by different types of chemical modifications. And so I've included a few examples down here in sort of the red shaded area, including things like acetylation, methylation, and some others. But I will just note that there are over 170 of these modifications that have been annotated, and the list is likely still growing. And so this is just a snapshot. These different types of RNAs I've listed here are also just a small snapshot, but I just want to highlight that different types of RNAs will carry very different complements and very different amounts of these different types of modifications. And so that really impacts sort of how modifications regulate their functions. And so in our lab, we're really interested in understanding how these different modifications really function at a very molecular level. And so in particular, we're interested in understanding on a single RNA molecule, how do these modifications alter subcellular localization, for instance, or RNA fate? Do they regulate specific binding proteins? And how do different types of modifications coexist, cooperate, or maybe even compete in cells? And it's not letting me advance my slides. 1 second. There we go. So today's talk is going to focus on one particular modification, N6-methyladenosine, or M6A, which is shown here on the left. And this has really been very prominent in the literature of late because it's been sort of heavily studied in its sort of a regulatory mechanisms of gene expression. And so it's installed co-transcriptionally by this methyltransferase complex. And then it can impact an array of functions in mRNA processing, including many of the nuclear processing steps that have to happen before an mRNA can be exported into the cytoplasm, and where it then also has impacts on things like mRNA stability and translation. And so given these pretty ubiquitous effects on an mRNA's life cycle, it's probably not surprising that M6A has also been implicated in a wide variety of cancers, and particularly M6A-mediated regulation of various oncogenes and tumor suppressors is known to have really critical impacts on sort of the progression of cancers, but also in treatment. And so for that reason, it's maybe not surprising that M6A modification in particular has been targeted therapeutically. And in, I think it was 2021, the Kouzarides, Celebes, and Roche labs published this first generation inhibitor of metal-3, which is the M6A methyltransferase. And this inhibitor has now undergone some medicinal chemistry development, and derivatives of this inhibitor are now being tested in clinical trials for a wide variety of solid tumors. And so it should also be noted that beyond just being sort of a therapeutic modality, it might also serve as sort of an adjuvant therapy. And so it's possible that by sort of modulating the M6A levels on different sort of key mRNAs in tumors, it might actually be able to sort of increase or decrease susceptibility to other treatments. And so this adjuvant therapy modality is being explored in the literature as well. But one thing I'll note about this, and this is something that sort of caught our eye, is this little annotation here, which is that in these clinical trials testing metal-3 inhibitors, nervous system tumors are always excluded. And this is apparently, from talking to my medical colleagues, pretty typical because many drugs have issues crossing the blood-brain barrier. But the reason this caught our attention is that we had, particularly through the Sontag Foundation support of our work, a developing interest in the lab in studying how RNA modifications might be regulating brain cancers. And so Emily Dangelmeyer in the lab, a graduate student in the lab, really spearheaded this effort to start our sort of foray into glioblastoma biology. And so she initiated this project with a really talented undergrad, Jessen, who's since moved on to medical school and is now joined by Nikki Harry, a postgraduate in the lab. And so glioblastoma, for those who are not aware, is a grade 4 glioma. It's really aggressive and it's the most commonly diagnosed primary brain tumor. Patient prognosis is quite poor. There's a very high recurrence rate of this disease, approaching 90% within 2 years. And this is in large part due to drug resistance. So drug resistance is very, very prevalent in this disease, but there are very limited treatment options, particularly once that resistance has arisen. And critically, this drug resistance isn't driven by DNA mutations. So in fact, it's not, it seems to be something more like at the RNA or transcriptional level, where we see evidence of widespread transcriptional changes, both that are heterogeneous, both within cells within a tumor and across different tumors. And so this is really an interesting sort of opportunity to look for RNA-mediated regulation, which I will note, in addition to sort of just brain cancer biology, the brain in general and sort of development and other aspects has been a really fascinating place for RNA biology of late, in particular M6A biology. And so this motivated us to learn a little bit more about this. And so just to sort of introduce you to the clinical challenge here, drug resistance is a huge challenge in GBM, as I mentioned. And so initially, patients will typically have surgery to remove as much of the tumor as possible. They will then undergo treatment with radiotherapy and this alkylating agent called temozolomide or TMZ. And then after a period of time, typically what we'll see is even despite adjuvant TMZ therapy, the tumors will sort of become resistant and recur. And so while the addition of this temozolomide chemotherapeutic certainly improves progression-free survival in patients, it's certainly not been a cure-all because we still have relatively poor sort of overall outcomes here. And so TMZ, as I mentioned, is an alkylating agent. And so it introduces alkylation to cellular nucleic acids, including DNA. And the particularly toxic species is O6-methylguanine on DNA, which induces DNA mismatch repair pathways. And so at that point, essentially, you can think of the outcomes sort of in terms of two extremes on a spectrum. And so on the one hand, the cell could undergo overwhelming DNA damage and essentially have insufficient repair capacity, at which case it just undergoes cell death and is therefore resistant to the TMZ therapy. But on the flip side, if a cell has very high DNA repair capacity, it can in fact sort of undo all of the damage that TMZ is doing and thereby sort of become resistant and survive. And so a key mediator of this, this whole process is this MGMT enzyme. So it's a methyltransferase that helps to sort of remove this damage. This is going to be a key player moving forward because MGMT is in fact clinically known to be a critical mediator of TMZ resistance as well. And so at baseline, the promoter of this MGMT genomic locus is typically methylated, and this sort of shuts down transcription. The MGMT mRNA is not expressed. And in fact, in many RNA-seq data sets, you will barely see any evidence of expression whatsoever. But if the promoter, becomes unmethylated, then we get upregulation of this MGMT mRNA, which then allows for really high levels of DNA repair capacity, at least in terms of this alkylation damage. And so the methylation status of the MGMT promoter is in fact a diagnostic that's used for TMZ sensitivity in the clinic. And so getting back to this question, we started to wonder, given all of the success that METL3 inhibition has had in other cancers, could targeting METL3 or even other RNA modifying enzymes potentially be a viable therapeutic strategy for drug resistance in GBM? But to start with, we're fundamentally a basic science lab. We really like tinkering with cells and molecules. We don't really deal with whole living organisms. And so we wanted a good model to be able to study in the lab. And so Emily embarked on this effort after sort of collating the literature on this topic to generate cell lines that were resistant to TMZ in the lab. And so we took glioma cell lines and then selected them with moderate doses of TMZ over the course of many weeks to then generate stably resistant clones. And of course, most cells will die early on, but then these stably resistant clones will persist and grow up over time. And so she undertook this first with this U87 MG cell line. And you can see that when you compare sort of the parental line and the TMZ resistant clones that grew up, that if you compare to sort of an untreated baseline, you can see that the parental cell lines, when acutely treated with TMZ, will start to die off, whereas the resistant clones are fine. And this is even more striking in a colony formation assay, where you can see in the parental line that the TMZ essentially obliterates these cells, whereas in the resistant clones, the TMZ treated and untreated are relatively indistinguishable. And so Emily went on to do this in multiple cell lines. And so we now have multiple paired cell lines of parental and resistant lines for these different cell lines so that we can sort of rigorously test what might be going on in terms of RNA modifications in multiple lines. So the first thing we did in sort of classic RNA lab fashion is we did some RNA-seq analysis. And so we just wanted to see what might be sort of generally upregulated and downregulated in the parental lines and the resistant lines. And we were quite heartened to see that MGMT, in fact, is the highest upregulated transcript by fold change. And so this was very heartening because this was quite literally the only molecular marker we had to look at in terms of sort of assessing resistance at a molecular level. So that was really good to see. And so we then went on to assess the MGMT levels, both at the RNA and protein levels in all of our different paired parental and resistant lines. And you can see in every case, we see a massive upregulation of MGMT at the RNA level. And you can see evidence for this at the protein level as well. And so the basal levels of MGMT are a little bit different if you sort of look at these two different exposures. But in every case, we see upregulation of MGMT. And so these, at least to the extent that we can assess, seem to be mimicking what is happening in the clinic. So the next question was, of course, well, what happens when we treat these cells with the metal-3 inhibitor? And so these are colony formation assays for all of our different paired and resistant, parental and resistant cell lines. And you can see that the end result is the same. And in every case, the metal-3 inhibitor basically kills the cells off pretty effectively. And I'll just add that we added a cell line down here, this T98G cell line, which is a cell line that's actually already resistant to TMZ. So we didn't have to make a resistant line for that. Now, if you look at this by a fluorescence-based cell viability assay, you can actually see, despite the sort of chaos of all of the overlaid curves, if you just look at the IC50 values for metal-3 inhibition that I've noted on the side here, there's this sort of very small but reproducible effect where we see a slightly lower IC50 in the resistant lines relative to the parental lines, which was kind of interesting because it suggested that there might be some differences in M6A methylated things in these cells that might be impacting their sensitivity. But the question, of course, is what is actually methylated in these cells? And so to look at this, we did an ECLIP experiment, or an enhanced cross-linking assisted immunoprecipitation experiment with an M6A antibody. And so in this experiment, we're basically taking RNA from these cells, either treated or not with the metal-3 inhibitor, then fragment and bind to an antibody and cross-link, and then essentially sequence what was bound to that M6A antibody. And because of that cross-linking, sort of map M6A sites at relatively high resolution. And so I'm just showing you some read pileups here that we sort of immediately drew our attention, which was in the MGMT locus. And you can see that relative to the input, we see a pretty good pileup of reads in the M6A ECLIPS sample. And importantly, that goes down when the cells have been treated with the metal-3 inhibitor. And so that suggests that this is really bona fide metal-3 mediated M6A methylation of MGMT itself. And I will note that this is a very sort of typical location for M6A, sort of around the 3' end of a coding sequence and into the 3' UTR. And so we were really quite confident that we had found this methylation on MGMT, as well as, I will say, many other transcripts, though I'm not really going to get into that today. And so just to sort of give you some confidence that this isn't just a cell line specific thing, we also by qPCR checked our other cell lines, and you can see that the MGMT is enriched in the M6AIP samples relative to the input in black here at comparable levels to a control MELAT1 here. So we're pretty confident that this is true across all of our different cell lines. But the real question, of course, is what happens to MGMT expression when you inhibit M6A methylation. And so by Western blot, we could see that treatment with the metal-3 inhibitor actually reduced the levels of MGMT in all of our different resistant cell lines. Again, the basal levels of MGMT are a little bit different, but we see a consistent drop in every case. And this is really interesting because the decrease in MGMT expression actually suggests that the M6A is stabilizing the RNA, right? So if we're removing the M6A and it's getting less stable, it's sort of a double negative here. And importantly, this is happening at the RNA level because when you check the RNA levels under these same treatment conditions, you do see that metal-3 inhibition is reducing the mRNA expression levels of MGMT as well. And so if you think about sort of the literature on M6A mediated regulation of mRNA stability, you can see that there's sort of two pathways I've listed here, but if you follow the literature at all, you may be more familiar with this one, which is this mRNA decay mechanism by which the M6A on mRNA is recruiting this binding protein YTH DF2, and thereby recruiting deadenylation and decay machinery. And this has been implicated in a wide variety of biological systems and is sort of a very clear function for M6A in a lot of contexts. But there is another alternative that has been shown in a few different contexts, including AML, which is that IGF-2BP proteins can bind to M6A and actually have the opposite effect, which is to stabilize them. And so we wondered whether M6A might be affecting stability of MGMT, but unfortunately, or fortunately, surprisingly, MGMT is very, very stable for an mRNA. And it turned out that the traditional sort of transcription, inhibition, and decay assays were not super helpful to look at this because it was such a long-lived mRNA. And so instead, we turned to a pulse chase format where we use phenyluridine labeling. We use a short labeling time and then collect RNA over many time points and then look at the decay of that labeled pool of RNA that way. And sure enough, you can see that when you treat with the metal-free inhibitor, you do see a reduction in the stability of this mRNA. And so our model is that there's this stabilization taking place. And so, of course, we were curious if this was actually mediated through the IGF-2 BPs. And sure enough, at least preliminarily, we can see that knockdown of IGF-2 BP2 does seem to reduce the levels of MGMT at the protein level. So we do think that this is, at least in part, explaining sort of why M6A is stabilizing this mRNA. But we were curious, just for a last bit of data here, to see whether this is exclusive to MGMT or not. And so we turned back to our RNA-seq analysis, which actually didn't mention before, we did with and without metal-3 inhibition. And so when you compare sort of wild type and resistant cells with and without metal-3 inhibition, you can see that most of the transcripts that change with the M6A inhibitor are shared between the two cells. And so it's not that M6A mediated regulation is being totally rewired. Most of these things are sort of the same. But there are many transcripts that change upon metal-3 inhibition that go up, which is what you would expect for the sort of canonical M6A mediated decay mechanism. But what was pretty interesting was that if you sort of subset on the resistant specific transcripts, so things that were really upregulated in the resistant cells, those transcripts did tend to go down with the inhibitor treatment, suggesting sort of that they were being regulated in a manner similar to MGMT. And that's something interesting that we're still following up on to see if that might potentially have something to do with either drug resistance mechanisms or other important transcripts as well. So just to wrap up, so I've shown you today that we've identified sort of a really interesting and quite stable mRNA that is very critical for GBM, which is MGMT. And we think that it's being stabilized by M6A through this IGF-2 BP binding protein mechanism. But there are, of course, a lot of questions that remain to be answered, including sort of why is IGF-2 BP binding and not a YTH protein? And this is still sort of a bit of a puzzle because the sequences are really quite similar in terms of the preference of these two proteins. We suspect that there's maybe a bit of feedback that's actually altering the balance of expression of these two families of proteins, but that's still something we're following up on. And then more broadly, of course, we're really interested in understanding and working with more sort of clinically oriented folks to see if maybe this will actually be therapeutically useful and actionable in the future. And so with that, I'd just like to wrap up and thank my fantastic lab. As I mentioned before, this work was spearheaded by Emily, but has been sort of a collaborative effort, not just with Nikki and Jessen, but with everyone in the lab. I'd also like to thank my fantastic collaborators, including many at the RNA Center, which is just a wonderful community. And then my funding sources and a disclosure that I'll just note here. And with that, I'd be happy to take any questions. Thank you.
Great. Thank you so much, Siggy. I really enjoyed your talk. Maybe I'll start looking for your questions to show up in the Q&A, not in the chat, please. So please post questions in the Q&A. So I Can you hear me talking? Oh,
Yes.
great. I just got some weird alert. Anyway, I wanted to ask about the pull down experiment where you identified the 3' UTR as having the M6A modifications.
Yeah.
It seemed like the signal was spilling over into the intron. Of course, I would notice that. And I wondered if you think that's significant, or is that just a misannotated 3' last exon?
It could be either. I will say that I have gotten this question a few times, and we need to look at this more closely because one thing, the reason I zoomed in on that region is MGMT is absolutely massive. It has huge introns, like way more intronic sequence than exonic. And I do sort of wonder if there's an element of
Mm,
this that has to do with splicing regulation.
hmm.
And maybe an intron that's a bit slower or faster, who knows? And maybe
Right.
the M6A is having something to do with that. So that's something we still need to look into, probably by long read. And turns out I know a person who's quite good with long read sequencing. So it's an interesting question, but we haven't looked at it yet.
Cool. All right. So a couple of questions have come in. The first one is from Norman Chu, and he's asking, have you looked at other RNA modifications that might coexist on the same MGMT transcripts?
Yes, so that is something that we have actively tried to do. One sort of clear choice was to look for editing sites, actually, because RNA editing has also been implicated in GBM, and thus far we haven't seen any. Using some tools that we're developing in the lab to look for other modifications, we actually did do some sort of targeted sequencing of the 3' different regions of the 3' UTR to see if we could find any hints. So far, stunning silence and nothing, but that doesn't exclude the possibility that they're elsewhere. So we are going to look a little bit more.
Okay, I just typed your answer. Okay, this is from Cece or Chris Kahn. Great talk for the RNA-seq. How long did you treat with STM? And did you check the level of M6A altered by mass spec or ELISA, et cetera? If so, how much was lost in the time treatment?
Yeah, this is a good question. And I left the slide out. Maybe I should have left it in. So we did check by mass spec because we wanted to find sort of a time and a dose of the M6A inhibitor that reduces M6A but doesn't kill everything, which is, a very fine line. And so we ended up settling on a 50 micromolar treatment for about 96 hours as being pretty consistent. We lost, I'd have to go back and look. It was maybe about 75% of the M6A by mass spec. So not completely gone, but again, also trying not to kill the cells. And so, yeah, we did assess that by mass spec to look at sort of the bulk level M6A changes.
Cool. Do you know, this is Pedro Batista, do you know the IDH status in these cell lines? Do you know if there's an accumulation of 2-HG potentially inhibiting demethylase activity?
Yes. So this is a super interesting direction that we have not yet pursued. These are IDH wild type. But we would be super interested to look at what happens, in sort of the IDH sort of mutant cases, particularly given the effects of 2-HG on some of these enzymes. So these are wild type, but I think it would be interesting to look at IDH mutant cells as well.
Great. I'm typing this. I don't know if I should type this. Okay. Um,
All right.
so let's see. I
I
don't
think we
see
need
more
to hand
questions.
it
I think we're
over.
at like 29 and a half minutes anyway, so maybe that's
Yep.
perfect.
Yeah.
Um, so we could pass our baton to Frank in that case.
All right. Well, great. Thanks to Siggy and Karla. Fantastic talk. So welcome to the talk from the HIRM today. It's a pleasure for me to introduce Dr. Alex Wesselhoeft to you. Alex earned his PhD at MIT, where he created a novel RNA circularization technology that led to a number of key publications, as well as him co-founding a company called Therapeutics, which has led the sort of space in circular RNA for therapeutics. He directed RNA research there and development for several years, and then has more recently come back to academia, where he joined the Gene and Cell Therapy Institute at MGH to build a circular RNA manufacturing core and research lab. He also has an academic appointment at Harvard Medical School as an instructor in the Division of Engineering and Medicine at Brigham and Women's Hospital. So it's wonderful for us to have Alex join us today. So Alex, go ahead and tell us all about circular RNA.
Great. Thank you, Frank, and very excited to be here today. Let me just go ahead and share my screen here. All right. Okay, does this look right?
Yes, looks good.
All right, perfect. All right. Yeah, so today I'm going to talk a little bit about circular RNA and the work that I've been involved with over the course of several years at this point, and covering the sort of foundations of circular RNA as a potential therapeutic technology back when I was working on it during my PhD in the Anderson Lab of MIT. And then I'll talk a little bit about how we engineer these things, how we manufacture them at scale, some of the challenges that face these sort of two efforts, and then some of the early medical applications of circular RNA that are being explored, both in academia and in industry. So of course, I probably don't have to tell anyone here that RNA has been gaining ground in terms of its use as both research tools and therapeutics. On the research side, of course, there's been a sort of resurgence of RNA manufacturing capabilities from small RNAs to large protein coding mRNAs that has really rapidly developed, especially over the last 4 or 5 years. On the back of the approval of the mRNA-based coronavirus vaccines, which out of Moderna and Pfizer, which are the first approved mRNA drugs that have been generated so far. And of course, mRNA and RNA in general has a lot of advantages over more historically used gene delivery vectors or gene manipulation vectors like adenoviruses or adeno-associated viruses, in that you have more flexibility with redosing, you have more control over the therapy. It's sort of a hybrid between a virus and a small molecule in some ways. And it also has really good developability. So you can basically manufacture these things without requiring cells. But there are significant challenges in translating RNA technology into a medicine. We've seen some of those challenges overcome recently with the coronavirus vaccines. But in terms of therapeutic applications, vaccines are actually probably one of the easiest applications you can go after, since you only need a teeny tiny amount of antigen expressed in the target tissue, and you can do a small local injection to stimulate a powerful immune response. So this is very much a first step towards mRNA therapeutics, but the broader application of mRNA into other therapeutic areas, such as immuno-oncology, autoimmunity, and rare diseases with protein replacement, is still pretty limited, and there's been nothing that's been approved there so far. And one of the key limitations is the instability of the mRNA itself. And so that's where this idea of RNA circularization came from, was as a topological solution to stabilizing mRNA. And so mRNA is typically degraded through by exonucleases, decapping enzymes that chew up the RNA from the 5' and 3' ends. And most of the RNAs within eukaryotic mammalian cells are degraded by exonucleases. This exosome complex shown on the right here. And so the idea is, if we can remove the termini that are used by the exonuclease, the exosome complex, to degrade the RNA, then we can potentially stabilize the structure. And that stabilization could lead to a longer duration of therapeutic effect. And you get more protein per RNA, and then you get a dosing advantage, a lower redosing need, and a better therapeutic index. And the hope is that a better therapeutic index with this underlying platform technology will open up new doors that are not currently accessible to mRNA therapeutics. So how can we make circular RNA? That's the idea and concept that I was starting with back in 2016. And that brings me to my first section, which is how do we engineer circular RNAs? So when I was starting working on this, circular RNAs had actually been made before. There were some very small circular RNAs that had been made by some groups in the 90s. And on the left, you can see a gel where you can see a precursor RNA band and then 124 nucleotide circular RNA band. And this material was made through an intron-mediated back splicing process, which is why you have these intron fragments down below here. More on that later. But you can see that even for this very small circular RNA, you still have a lot of precursor left over. So the circularization efficiency is not very good. And the bigger the circular RNA gets, the harder it is to circularize. So by the time you get to something that can actually encode a protein, like a 1.5 kilobase RNA, which is about as small as it gets, since you need an IRES in there, these things become quite challenging to circularize. And enzymatic ligation methods are just not quite good enough to make these circular RNAs reliable. So that was one challenge that needed to be solved. Second challenge was, can we translate protein out of circular RNAs? People hadn't really made them, so we didn't really know whether we could translate protein out of them. There was a big question in the field of natural circular RNAs, which are different from these synthetic circular RNAs, of whether these natural circular RNAs in Drosophila brains and so on were translated into small peptides. So that was still an open question in the field at the time. And then finally, therapeutics. Circular RNAs, of course, had not been used in any sort of preclinical or clinical context. One of the first things we did was engineer a system for efficient circularization of RNA precursors in vitro. And we used this permuted group 1 intron system, where we took a natural ribozyme, we split it in half. We took the second half, put it in front of the first half, and then inserted a region of interest in between the junction. And in this case, we have an EMCB IRES and a Gaussian luciferase coding region, which is a fairly small protein. And theoretically, this should go through the standard dual transesterification process of a group 1 intron and yield circular RNA and free introns. And long story short, it didn't work the first time we tried it with this cargo. But after engineering a bunch of these, we call them accessory elements, including spacers to help elements fold properly, but also internal duplex forming regions or homology arms, and external duplex regions or homology arms to help bring those intron fragments together over a long distance. We were able to show that we could circularize a wide range of constructs very efficiently, all the way up through Cas9 was the largest one we did in this publication from 2018. So the red arrows here are pointing to the circular RNA product. And you can see that compared to the precursor RNA, which is the fainter band below the circular RNA product, most of the material in these reactions was circular RNA. But the larger you get still for Cas9, you still see a drop-off in efficiency. So once we could make circular RNA, we needed to figure out how to translate protein from circular RNA. And of course, with mRNA, you have this cap structure at the 5' end that initiates translation. But if you don't have any ends, then you can't really have any caps, although there are some recent publications that would suggest that you could sort of non-covalently or covalently, but on a different hydroxyl group, cap circular RNA as well, and it sort of does work to some extent. In trying to figure out how to get translation in a cap-independent manner, we at the time had this really great example of nature's way of doing this, which is this sequence element called the internal ribosome entry site. And this is a sequence element that's about 500 to 700 nucleotides in length. It's highly structured, and it promotes cap-independent translation on viral transcripts. So what we did was we took this IRES, the one that people typically use is the EMCB IRES from the EMCB virus. We put it into our circular RNA, made circular RNA, and then we found that indeed the IRES was capable of driving translation in circular RNA. So that was very exciting. there are many IRESs out there, and it's a highly unexplored space in circular RNA. So we wanted to assess a broader range of IRESs and see if we could further boost translation from circular RNA by modifying the IRES element and choosing IRES elements that have high potency or efficiency. And so what we did, and this is work that happened that I led while at RNA Therapeutics, was we designed a library of 3,000, or computationally designed a library of 3,000 IRES candidates based on natural sequences. We generated DNA templates for each one individually. So this was a massive arrayed screen, many different plates, 96-well plates. We generated purified circular RNA and then formulated the circular RNA into lipid nanoparticles. And then we tested the formulated circular RNA in primary human cells, including T cells, myotubes, and hepatocytes. And what we found was that there's a huge range of translation efficiency that arises from these IRES elements when placed into circular RNA. And so these graphs just show the expression of the top 1,000 IRESs in myotubes, hepatocytes, and T cells. Each dot is an IRES. And as you can see, the cloning vector EMCB is actually not a great IRES. We were finding IRESs that had 40-fold higher potency than the EMCB IRES in these primary human cell types. So this was very exciting to find that the IRES was such a massive and significant contributor to translation in circular RNA, and that there are some IRESs out there that just haven't even been characterized at all, that are way, way better than the typical EMCB IRES. So in order to make a drug, of course, you need to manufacture these materials. And circular RNA has maybe some unique challenges compared to mRNA, because mRNA, of course, can be modified. And typically for therapeutic applications, the modifications used are uridine modifications, like 1-methyl-C-uridine. And these uridine modifications reduce the immunotoxicity of the RNA and reduce the amount of purification that you need in order to have a clean product, basically. And unfortunately, with circular RNA, that can't be done. You can't modify circular RNA because it massively changes the structure of the IRES and the structure of the introns that are used to circularize it. And so if you tried to make modified circular RNA, which is something that we published on, it basically doesn't work anymore. So we needed to figure out ways to purify the material so it wasn't immunotoxic without using these modifications. And we developed this very onerous process using a combination of RNase-R, HPLC, and a phosphatase treatment to basically bring the immunotoxicity of circular RNA back up to baseline. And you can see in these charts that if you do all of these purification methods together, you get a really nice stability curve of expression in A549 cells, which are very sensitive cells. Your cytokines go down to baseline. And if you look at TLR responsive cells, your circular RNA looks basically exactly the same as this modified linear mRNA. While at Orna, I also led an effort to condense this very onerous manufacturing process down into a simple two-step manufacturing process, whereby we could start with something that looks like the chromatogram on the left and with something that looks like the chromatogram on the right, with a little bit of nicked RNA remaining over, but the vast majority of it is intact circular RNA. And this manufacturing process yielded about 40 to 60% total process yield, and we scaled it up beyond 100+ milligrams, which is enough for a small human vaccine clinical trial, and certainly enough for non-human primate studies. When we looked in vivo at the immunotoxicity of these materials, here's a TNF alpha in mice, looking at a comparison of unmodified and modified mRNA versus a standard processed circRNA versus our new ultra-pure processed circular RNA. You can see that the TNF alpha response to circular RNA basically looks like PBS. It's actually even lower than the 5-methoxy-uridine modified mRNA, and certainly a lot lower than the unmodified mRNA. Due to this enhanced purification process that we had. And so now at the Gene and Cell Therapy Institute of MGB, One of my goals is to basically democratize this technology that I've been working on for the last 8 years or so. And I've constructed the RNA Therapeutics core, which has been in business since September of last year. So the core is now open, and the core sells stock circular RNAs encoding a variety of different proteins, reported proteins like GFP, mCherry, and luciferase. But we also make custom circular RNAs that can basically encode whatever protein you want, up to 10,000 nucleotides. We have a proprietary codon optimization algorithm that we've developed just for circular RNA and can make things high throughput. And finally, we also do lipid nanoparticle formulation in a variety of different standard lipid nanoparticles and some custom RNA therapeutics core specific LNP formulations. So if you guys are interested in circular RNA or formulated circular RNA for research purposes, please don't hesitate to email the core. The email is down below here, rnatherapeutics@mgb.org. And here's some examples of what the material is that the core is making. So on the left, we have a circular RNA chromatogram. You can see again, you have some concatenations and nicked material down here, but it's the vast majority of it is intact monomeric circular RNA. This is 2.5 kilobases, so reasonably large, not huge. I'd say probably about average. And then when we transfect these into cells, sensitive cells, we see essentially no IL-6 response, no interferon beta response. The material itself, compared to mRNA, is stable and potent. So the green lines here are circular RNA, whereas the gray lines are modified mRNA. So you can see that, and this really showcases the stability of circular RNA, at least in these cell lines, where you can see that over 6 days, you get an accumulation of EGFP signal after transfection of these cells with circular RNA. Whereas if you do modified mRNA, you get a little bit of signal at the early time point, and it sort of accumulates a little bit at the second day, but after that, it basically flatlines. And so even out to 6 days, you're getting good translation from circular RNA. If you formulate these materials into lipid nanoparticles and transfect different cell types using a different reporter, firefly luciferase, which has a half-life of 2 hours, you can still see the difference in potency comparing circular RNA and mRNA as gene expression technologies. We've also been hard at work making really big circular RNAs. So here's 2 examples of some really big circular RNAs that we've been making. The left is a PEmax prime editor, which is about 7.8 kilobases, so fairly large. And this is editing in these KOLF2 cell line, where we see that we only need about 4 micrograms of circular RNA to edit cells as much as you need 16 micrograms of mRNA. So it's about 4 times, actually it's over 4 times more potent than the mRNA editor. And on the right, this is an even larger circular RNA. It's 9 kilobases. It encodes the full-length protein NF1, where we see complete gene replacement of NF1 in these NF1 knockout Schwann cells. In addition, we're pushing out some new technologies that are becoming available through the RTC, RNA Therapeutics core. So we already have our proprietary codon optimization algorithm in place, which significantly improves the stability of these circular RNA constructs without compromising peak expression. We're also developing new circularization systems to further improve our ability to make really big circular RNAs. And we're developing some unique biodegradable lipids that are showing some early promise in vitro on the left and in vivo on the right. We've also generated some really interesting data on delivering circular RNA to a range of tissues with different partner labs that we work with. So in all these cases, circular RNA is being formulated into lipid nanoparticles and tested in a variety of different delivery routes. So the first is intravenous, where we see great expression in both liver and spleen using an RTC formulation compared to ALC 0315, which is the formulation used by Pfizer in their vaccine. With intravitreal injection, actually, we're seeing really interesting results with our RTC 002 formulation, showing potentially, we're trying to still figure this out, but potentially photoreceptor expression with the RTC002 formulation, and this is circular RNA that's encoding the GFP. And then on the right is a lung delivery example using a variant of our RTC002 formulation after intratracheal delivery, where we get robust circular RNA expression in lung epithelial cells. So I see I'm sort of a little bit short on time here. So there are a number of medical applications that people are looking into with circular RNA, and all of this is work that I've either collaborated on recently or led while I was at Orna. So as a vaccine platform, circular RNA can be used very similarly to mRNA. So this is a coronavirus vaccine that we made while at Orna Therapeutics, and we found that it generated very strong binding antibody response in mice, and with essentially zero optimization. So this was quite exciting. We've also looked into circular RNA as a rare genetic disease platform. So using circular RNA as protein replacement. In this case, we're encoding the extremely large protein dystrophin, which generates an almost 12 kilobase long circular RNA, which is quite challenging to make. But we are able to see full-length dystrophin protein generated from the circular RNA after translation in a cell-free assay on the top. And on the bottom, we see translation of full-length dystrophin circular RNA in primary human myotubes. And probably the most active area of research with circular RNA, and currently mRNA as well, is the concept of in vivo CAR T, where you basically take a chimeric antigen receptor, you encode it in a circular RNA, you formulate that circular RNA in an LNP, you deliver it to T cells in vivo as an injectable drug. Those T cells become CAR T cells, and then those CAR T cells will kill their target cells. So on the right is some data showing the outcome of this process, where with a HER2 targeting CAR in a B-cell lymphoma model, you can see that these mice basically succumb to lymphoma by day 25, and the lymphoma is basically all over the body. After treatment with an LNP circular RNA at 0.3 mpk, the lymphoma is controlled after a single dose and eradicated after 2 doses. And you can see these mice, with these mice, they basically just keep going through day 56. So that's my sort of broad summary of circular RNA. We have engineering, manufacturing, medicine. We figured out how to circularize large RNAs from the smaller protein coding RNAs like eGFP encoding all the way up to something that can code full-length dystrophin. We figured out expression via IRAS sequences and identified some novel IRAS sequences from nature in a massive parallel screen. We developed a manufacturing system that resulted in a simple 2-step synthesis and non-enzymatic purification process that yields high-quality material that does not stimulate immune response in vivo. And then we started to look at a variety of different medical applications of the technologies, including vaccines, CAR T, and protein replacement therapy. So with that, I'd like to thank my lab, and who's been with me for the last 2 years now while I've been at GCTI and doing some really exciting work on translational medicine. The RNA Therapeutics core, who make excellent high-quality circular RNA and formulations and have really promoted tons of collaborations within the MGB system and with other academic institutes. And of course, the GCTI team, who has supported the development of the RNA Therapeutics core and the research lab. And so with that, I'll take any questions.
Yeah, wonderful. Thank you, Alex. That was fantastic. We already have a bunch of questions in the Q&A, so I'll just get started right away and remind everybody, please type your questions there. First question from Amit Rai says, Alex, nice talk. Can you please give us your insights about the subset of RNA ligases which you think could play a vital role in circular RNA biogenesis in vivo?
Yeah, so in vivo, so ligases typically don't circularize RNA in vivo, I would say. It's certainly been demonstrated to be possible. I think RTCB ligase is the ligase of choice there. And there are some people applying it therapeutically via AAV-mediated delivery of small RNAs that can sort of self-cleave and then ligate using these endogenous ligases. But most circular RNA is generated through the process of backsplicing and not necessarily an enzyme-mediated ligation process.
Great. Next question is from Jean Petrella. When comparing mRNA versus circular RNA for immunogenicity, toxicity, were identical coding sequences used?
So yeah, that has been done before, yes. And I think in that experiment, yes, the identical coding sequences were used. But I will say that the codon optimization that you want for circular RNA versus mRNA is not the same. Because you have special considerations for circular RNA. And one of the things that we think about when we develop the codon optimization algorithm is how to promote circularization, which means bringing the ends together through design of the coding region, which is something you don't have to really worry about with mRNA.
Yeah, cool, thanks. Larry Melidis asks, in the cases where the IRA sequences did not initiate translation, do you feel like you're getting a change in the IRA structure? And how do you optimize the IRS for the CDS or validate the structure in cells?
Yeah, it's a great question. So there's definitely some level of interference with between the IRES and the coding sequence as sequences that are next to each other, and of course you can get crosstalk between structures. There are IRESs that are more sensitive to this, and there are some that are less sensitive to this. For example, IRESs with scanning tracts tend to be a little bit less sensitive to neighboring regions because the scanning tract is just kind of a filler sequence anyway. In terms of determining the structure of an iris in vivo, you always have the option to do shape. But I would say, it's sort of okay. You can figure out maybe what's occluded, but you don't know whether it's occluded by RNA structures or by interactions with proteins. So that can be a little bit challenging to study the actual structure of what this looks like in vivo. Yeah.
Okay, cool. Timur Vaniski asks, very exciting progress. Can you rank the payload expression from circular RNAs versus modified RNA versus self-amplifying RNA by protein expression levels and RNA protein decay over time?
Yeah. So generally, I would say 9 times out of 10, circular RNA has a higher level expression and a longer duration of expression compared to modified mRNA. Compared to self-amplifying RNA, you typically have a much lower upfront protein production or protein translation because you start with less, but the RNA will amplify over time, and then your peak expression a few days later will usually be way higher than both mRNA and circular RNA. Of course, that comes at the cost of it being immunotoxic because you're going through a long double-stranded intermediate, and it's that long double-stranded intermediate that eventually shuts down the expression of the material. So you have to go through the immune response in order for it to stop replicating, basically. It's basically a virus. So I think, in terms of just expression level, self-amplifying RNA always wins in my view every single time. But therapeutic application-wise, I would say self-amplifying RNA is probably going to be limited to vaccines because of the immunotoxicity problem.
Right. Thanks. Anonymous attendee asks, first of all, nice talk. How long after injection have you followed the reporter signal presence and safety after injection in the retina and in other tissues? But I think maybe answer for the retina.
Retina, very early, I would say, we have no additional information on that. So that's still TBD. But in other tissues, the duration of expression varies highly depending on both the protein, which will have its own stability associated with it, but also the RNA turnover can be faster or slower in different tissues. Typically, I see usually a 3 to 5 times improvement in half-life compared to a modified mRNA. But, you're starting at like 12 hours, so 3 to 5 fold. You're at like, 2 days, 3 days, 4 days, something like that, for the circular RNA.
Great. Margaret Bome asks, well, first of all, great talk. Have you compared the encapsulation efficiency of the LNPs using linear versus circular RNA?
Yeah, the overall, the circular RNA actually formulates better than mRNA because you don't have that really unstructured poly-A tail. So that typically leads to higher encapsulation efficiencies, which are admittedly also very good for mRNA, 95% plus, usually depending on the LNP that you use. But also the size of the particle tends to be a little bit more compact with circular RNA.
Great. And then our last question, because we're getting a little over time, is from Shorty Agishi. Asks, can you discuss any considerations for circularizing non-coding RNAs like guide RNAs?
Yeah, so a small RNA like a guide, I would say you can chemically synthesize it. You can add tons of crazy modifications to it. So I'm not sure if it's worth circularizing a guide RNA for the purposes of stability. It seems like maybe more effort than it's worth. Where circular RNA really shines is beyond the scope of chemical synthesis. So like after you get 120 nucleotides, I think where chemical synthesis really falls off, that's where you might want to really consider circularizing a non-coding RNA. And actually, I would say non-coding RNA may benefit from circularization even more than coding RNA, because at least with coding RNA, you've got a cap and a tail protecting you. But if you want a long non-coding RNA without cap, for example, that thing is going to be very sensitive to decay. And so circularization could be a massive benefit there.
All right. Well, thank you very much to both of our speakers today. I'm going to close out just by reminding everybody that we'll have our next RNA Collaborative seminar series on Wednesday, July 9th at 4 o'clock Eastern Time. And it'll be MD Anderson Center for RNA Interference and Non-Coding RNAs and the UCSC Center for Molecular Biology of RNA. So please join us next month. And once again, thanks to all of our speakers. It was a fantastic set of talks, and all the best to everybody. And we'll see you next month. Bye-bye.
Thank you.
Thanks, Alex and Siggy.